We'll give it one more minute and then we'll get started. Okay, welcome everyone to the New Mexico Tech Biology Department's panel on the COVID-19 vaccines. And here we hope to give you some accurate information that you can use to make decisions and potentially help other people make decisions in your life. And I'm really excited about the panelists that we have today to talk to everybody. So I'm going to give a brief introduction to our panelists and then let them take it away. So today our panel will include a history of vaccine and vaccination campaigns in the United States and somewhat around the world. And that will be done by Dr. Tom Kieft. Dr. Kieft is an environmental microbiologist who has also had clinical lab experience, and he teaches a course in medical microbiology and immunology here at New Mexico Tech. Dr. Kieft completed his master's degree at New Mexico Highlands University, his PhD at University of New Mexico. He did a postdoc at UC Berkeley, and then he joined the New Mexico Tech faculty after that, and he's been here ever since. So he's a really great authority in microbiology, and we're excited to have him here. Thanks, Dr. Kieft, for joining us. After the vaccine sort of historical perspective, we're going to hear about the molecular biology and the biotechnology of how these vaccines work, which is really exciting science. I think also potentially a source of concern for some people out there. So we want to give you the biology behind that and talk about the technology itself. And Dr. Snezna Rogelj will give that overview. Dr. Snezna received her bachelor's in physics from The Ohio State University and got her PhD in biochemistry from Boston University School of Medicine. She did a 5-year postdoc in cancer research at MIT and Harvard, where she studied parasites. And then after that, she studied parasites in India and immune cell interactions at the University of New Mexico. After that, she joined the New Mexico Tech faculty and has been here for the past 22 years. And now she's a full professor here. She was the department chair for 8 years in the biology department and helped establish the biomedical sciences program, of which many of you are partaking, as well as the biotechnology PhD program, which is growing. And her research is interdisciplinary, but has focused over the development of anti-cancer, antibacterial, and anti-parasitic drugs. And she has trained many undergraduates and graduate students in the last, hundreds of undergraduates in the last 10 years, and over 40 biology master's students and 1 biotechnology PhD student in research methods and design. So we're very excited to have Dr. Rogelj talk to us about the molecular biology of the vaccine. And I think you'll learn a lot. I certainly am excited for it. After Dr. Rogelj's introduction to the biology, we're going to hear from Dr. Bryce Chakerian about the FDA approval process and why this vaccine was approved so rapidly, or seemingly rapidly, as it were. So Dr. Chakerian is the Jeffrey Michael Gorvetzian Endowed Professor of Biomedical Research Excellence and Vice Chair of the Department of Molecular Genetics and Microbiology at UNM. He got his bachelor's degree from Berkeley and his PhD from University of Washington. And so we're excited for Dr. Chakerian. He works in development of some of these mRNA vaccines, in fact. And so he can speak a lot to the technology and the FDA approval process for vaccine use. Next, Dr. Danielle Turner, a postdoc in the biology department now, and also a medical student. She's double dipping here at the University of New Mexico. She is going to talk about the vaccine safety and efficacy data and how we can be confident in using this vaccine. Dr. Turner received her Bachelor's of Science and Master of Science in Biology and her PhD in Biotechnology from New Mexico Tech. So she's a Tech alum. And during her time as a postdoc at New Mexico Tech with Dr. Rogelj, she participated in the development of novel anti-cancer, antibacterial, and anti-parasitic drugs. She's immersed herself into the realm of drug entrepreneurship and has successfully licensed and commercialized a novel antimicrobial material with anti-SARS-CoV-2 properties. Dr. Turner served as the interim COVID-19 testing coordinator here at Tech during 2020, and she is currently a medical student, as I mentioned, but she finds a lot of joy in the trials and tribulations associated with lab work. So welcome, panelists. We're really excited to have you, and we're excited to have all of our participants as well. Participants, before we begin, I'd just like to give an introduction about how this is going to work. So essentially, each of the panelists is going to give an introduction to their topic, and then we're going to have some commentary from the other panelists. And I will sprinkle in questions that we got through the pre-panel survey. And then if you have additional questions, you can put them into the Q&A section, and we will try to get to them as time allows. And for those questions that we don't get to, we'll follow up with you via email to try and answer those questions and make sure you have good information to leave with. So without further ado, I'd just like to welcome Dr. Tom Kieft, and where he's going to talk about vaccine history and historical campaigns in vaccination.
All right, thanks, Joel. I'm going to share my screen and brief PowerPoint slideshow. All right, so as Joel said, this is going to be a historical perspective. I'm going to talk about the success stories in vaccinology, and there really aren't any disaster stories. This has been a real boon to mankind. So I'm going to show some graphs that show various incidences of disease with time. And so here's a lot of data on this one graph. The death rate from infectious disease in the United States over the 20th century. And obviously it's going down dramatically, and there are a number of factors in that. Water and wastewater treatment, antibiotics, just general hygiene, but vaccines account for a big part of this general drop in infectious disease, and are a big reason why we in the developed world enjoy such robust good health and relative freedom from infectious disease. Of course, there's another big feature in this graph, and that's the influenza pandemic. I'm not going to talk about flu vaccine especially, but that's one of the success stories too. The Centers for Disease Control and the World Health Organization monitor flu strains and generate vaccines that help to control the flu. And it could be argued that that's a big reason we haven't had another big spike like this. Of course, if we extend this into the current day, this graph, then there'd be a somewhat smaller spike, but a very significant one, 500,000 deaths in the US so far from COVID. All right, vaccines have been around for a long time, hundreds of years or more, and dating back to early days in China, India, Ottoman Empire. I thought I had another slide here. Let's see. Yeah, here's another version of this slide. The early vaccines might have been kind of a dicey proposition. One would buy the pox, they called it, from someone who would take smallpox pustule from one individual and give it to an uninfected individual to inoculate them. It may have been heat killed, may have been from mild cases. So it worked to some extent, and it spread to Britain and even the New World. Starting in 1721, it started being used in a fairly widespread way in Britain and New England. They called it variolation. But around about this time, they actually started collecting data to see, does it work? Dr. Turner will talk to us about how those data are used. Now, 1796, Edward Jenner, a doctor in the UK, is credited with figuring out to use a related virus. Of course, they didn't know it was a virus at that time, but pustules from cowpox that a dairymaid had. And he tested it in a young boy named James Phipps. And 2 months later, inoculated him with fresh, live smallpox pustules. Pretty risky thing to do, seemingly, but he had devised a really effective vaccine. And young James Phipps didn't get smallpox. And we can draw a direct line between Edward Jenner's vaccinia virus. That's where we get the name vaccination, by the way, vaca for cow. Anyway, we can draw a straight line from that to the eradication of smallpox worldwide in about 1977. It was officially declared in 1980. So a big success story. Probably none of you is vaccinated for smallpox. We don't have to vaccinate anyone for that anymore. All right. Another early vaccine, diphtheria, introduced in 1923. And here's one of these graphs where immediately following the introduction of the vaccine, there's a dramatic drop in the incidence of the disease. And so these days, everyone is vaccinated for diphtheria, very nearly everyone. And along with that, you're vaccinated for tetanus and whooping cough. And so, you know, we don't get those. Most people have bought into that. Another success story. Tetanus, by the way, if you contract tetanus and you're not protected by the tetanus vaccine, chances are you're going to die. Okay? That the incidence of that is extremely low for all three of these. So vaccines work. And now, one that I think take a little more personal interest in than some of the others, polio. I'm of an age that I remember some kids in school who were a little older than me who were on crutches. And I don't remember anyone in iron lungs, but this is actually a picture of one kid in an iron lung and a couple other kids in some other kind of device here. This is in my hometown of Sioux City, Iowa, in about 1952. Across the country in 1952, there were 60,000 cases, 3,000 deaths, mostly children, and thousands of cases of paralysis. Okay? And polio had been raging as an epidemic throughout most of the country the first half of the 20th century, peaking in 1952 and dropping off with the introduction of the Salk vaccine. And here are some kids in my hometown, I'm not in that picture, lining up and getting the flu vaccine. And about 6 years later, we all, polio vaccine. And 6 years later, we all lined up and took the Sabin oral vaccine. People bought into this. It was very widespread adoption of the vaccine. Nearly everyone got it. And now polio is nearly eradicated worldwide. The only cases are in the Indian subcontinent and small part of Africa, and probably within a year or two, due entirely to vaccination, polio is going to be the second infectious disease eliminated from the planet. Measles, another success case, introduction of the vaccine, a drop off in the incidence. In the 1950s and before, measles, mumps, rubella, chickenpox were thought of as childhood diseases that were rites of passage, and kids were encouraged to get it over with. Sometimes parents would send kids over to someone else's house where there was a case of measles to get it over with. Never mind that something like 3 to 5% of cases die. This is one that I hope you're all vaccinated with, this combined measles, mumps, rubella vaccine. And the incidence is very, very low, but this is one for which we have to maintain vigilance and make sure that people do comply and get vaccinated. There have been occasional outbreaks in communities that don't have enough of the population getting vaccinated. And this is as good a time as any to say, well, I'm talking about how I was around back in the old days here. People of my generation learned to question authority. And all this public health effort and vaccination is a government-sponsored enterprise. I still subscribe to the notion of questioning authority. And so you're perhaps questioning the vaccine, and that's why you're here for this panel presentation. I think you'll find in the case of vaccines, this history of vaccines and the current COVID vaccines, that this is one of the things that the government has gotten right. And we should all go along with it. All right. Oh, measles, mumps, rubella. People have heard of this story, and it's still sort of clinking around and used by some of the anti-vaxxers. Sorry. There was a 1998 paper in the journal Lancet that reported 8 children were diagnosed with autism within 1 month of receiving the MMR vaccine. This is a little more than 8 case studies, anecdotal data. You could probably find 8 children who were vaccinated and had all sorts of things happen to them within 1 month of receiving a vaccine. But it's of spurious correlation. Dozens of studies were done, retrospective studies looking back at incidents of these things, and then in real time prospective observational studies that totally debunked this link. The paper was retracted more than 10 years ago. And if you want more information, there's a very nice review on vaccines and autism and really the total lack of a connection. One more, chickenpox. The dates are a little more recent here. And chickenpox, high numbers until fairly recently. Things like hygiene, other methods of trying to control chickenpox didn't really do the trick. It's only when the vaccine was introduced, and now the incidence of that is very low. I'm going to skip over the COVID-19 vaccines because Dr. Rogelj is going to give an excellent presentation on this. I am, with my little history, going to bring us up to today. Okay? And so this is from a site called COVID Act Now. So far, 472,000 people in New Mexico have been vaccinated. And we're not reading in the papers, if you actually read the paper or seeing on the television news, about negative effects of that beyond a sore arm and things that Dr. Turner will talk about that are pretty minor. And our incidence is dropping. The vaccine is a pretty recent factor in this, but it's a really important factor to make sure that we don't have a resurgence. And with that, I will stop.
Thank you, Dr. Kieft. Really helpful introduction to vaccine history and everything. So first, I'd just like to open it up to the rest of the panel and see if there's any comments or thoughts that they would like to share on those topics, on historical vaccine topics. I know for me, I'm glad you brought up polio. My grandfather had polio, and he was actually precluded from fighting in World War II as a result of it. So it had long-term consequences. It wasn't just you get polio and either you die or you're okay. And it permanently disabled him for his entire life, such that he sort of limped around for all the time. Certainly that I knew him, but even in his mid-20s when he was supposed to be young and healthy. So it wasn't one of the, get it and you get it over with.
Right. And there's also post-polio syndrome. A college friend of mine, 2 years older than me, and so not, he didn't get the vaccine in time. He had polio, seemingly recovered fully. I used to play tennis and go skiing with him, very active man. He now has post-polio syndrome and can't do anything that active anymore.
And I think we're all kind of worried about that for COVID, right? Is that we don't really know the long-term consequences of this disease, of getting it. We do know that there are at least multiple month-long consequences that presumably could translate into multiple years-long consequences. I think we can talk about that later as well, but that's definitely one of the benefits of getting the vaccine, as opposed to, well, we'll just get it and I won't get sick, I'll be fine. You don't know that, because a lot of viruses like this can have long-term alternative consequences. Dr. Rogelj, can you unmute? I think you were about to say something.
You're muted, Snezna.
Yes, so I was just saying yes and confirming what you were, the point that you were making, that there are long-term consequences and they're not focused only on the elderly that predominantly die, but across the population, among the very young and middle-aged people as well. So that's a real concern that's associated with having gotten a full infection, and even upon a recovery, there are potential things to worry about.
Agreed. All right, well, with that, maybe that's a good transition to Dr. Rogelj, and she can tell us about the how the vaccines work to give you immunity, how this whole technology works.
Thank you, Joel, and hello, everyone. Let me find my screen. Okay, all right. So we all know that when we are speaking or singing, or coughing or sneezing, we expel droplets of our saliva. Droplets that with a good sneeze can go as many as 18 feet away from us and shower everyone around us. And that saliva will contain viral particles when we are infected with SARS-CoV-2. Now, those viral particles are tiny. They're maybe 100 times smaller in diameter than even the smallest of these droplets, aerosol droplets that tend to hang in the air sometimes for hours and be potentially inhaled by other people. So here is how one becomes infected by SARS-CoV-2. You inhale those droplets. They can be large, they can be small. If and when they contain the virus, they will be taken up into your nasal passages and into your lungs. As it turns out, the nasal passages are most susceptible to viral infection, but in either case, that virus finds a way to cells that line those tissues, that respiratory system, and they find what we call a receptor. They find proteins that are normally present on the surface of the covering of our respiratory system, and they bind to it and are naturally taken up into those cells where where the virus is essentially releases the messenger RNA, the code, which then allows that virus to replicate inside the cell and release many, many copies of themselves. Now, I want to point out at this point that when a virus infects these cells, in order to replicate, it needs to translate, it needs to make many different kinds of proteins that are essential for many different steps in viral replication, as well as proteins that ultimately make up the viruses that are released from the cell. But the sort of virologist will tell you that for every virus, you're likely to get something on the order of 100 viruses that are released from one cell, and those viruses literally go viral, they spread to other tissues, and because of the nature of their surface, viral surface, they can engage in tissues that express those specific receptors. And as it turns out, those specific receptors for this particular virus play a very important role in the entire body. So they're present throughout the body. So it's all these different tissues that are potentially infected with the viruses that are originally released just from a few cells within your respiratory system. And eventually they infect and affect a whole bunch of different organs throughout your body. And as a result of this infection, many things go wrong. I mean, the sort of the straightforward initial things that the patients observe is that they can no longer smell things right, they cannot taste them right, they might have diarrhea, but there are much deeper consequences of infection of all these different tissues. There are imbalances in the insulin production, imbalances in heart function. Blood volume is not properly regulated, and with the passage of time, those viruses, as they're replicating within each of those cells, within each of these organs, within each of these infected cells, cause damage, and the body fights back with initial inflammation. So you get this multi-organ injury, and I'm sure you've heard about the cytokine storm that is super damaging to all these organs. And as a result of that, people get sick. But when combined with pre-existing risk factors, meaning pre-existing illnesses or states of the patients, such as obesity and diabetes and hypertension and heart problems, the people really end up getting hit that much harder. This is data from 100,000 hospitalization. And all those pre-existing risk factors dramatically increase the chances of people dying. And people die. While the people who do get infected are mostly represented by those who are younger than 60 years. The ones who die tend to be 50 years or older. In fact, 95% of the Americans that were killed by COVID-19 were 50 years or older. So very disproportionate death rate that has a lot to do with other comorbidities, other problems that the elderly are likely to have. And the global cost of COVID-19 infections is exorbitant, right? Quite in addition to the personal cost, the societal cost. It has been guesstimated that the cost is something on the order of $16 trillion, that's $16 million around the globe, only a fraction of which probably would be the cost of initial prevention. But let's not go there. Let's see what your body is doing while the virus is replicating and infecting all these different kinds of tissues. Well, in addition to the initial innate response, sort of a general response to anything that's living and foreign, your body mounts a specific immune response. It's called an adaptive immune response. It's adaptive to the specific pathogen that is invading your body. And it is that immune response that takes much longer than your initial immediate innate immune response. So what I want to point out that initially upon infection, your body responds very quickly, but the adaptive to SARS-CoV-2 immune response takes time. It takes a couple of weeks, and that is really integral in development of a vaccine. And I'll be touching back upon that over and over again. So as we talk about the development of an adaptive immune response as a natural consequence of a viral infection. Okay, now what the body is doing is actually really complicated, and we don't have time to go through the details, but I want to just point out that this process, which takes initially a couple of weeks and can be amplified subsequently, is ultimately made up by cellular systems, immune systems, which produce antibodies, small molecules that can coat the virus before it has a chance to attach and infect another cell, as well as a population of whole cells that recognize that viral particle, that virus, and as it has infected a cell, and then cause that infected cell to commit suicide. So there's this one arm of the immune system that's both soluble and cellular, and then the other one that is made up of cells that are not necessarily actively carrying out either of these two activities, but sit to the side and wait for the second infection to take place. And as such, they're able to replicate very, very quickly and generate many cells that will attack infected cells, as well as generate large quantities of antibodies that can stop the virus before it can spread throughout the body. So you have this multifaceted, multi-armed immune response, and that immune response is takes time to develop. However, it also provides for lasting immunity. So when we do, which can be represented like this. So when we do vaccinate, what we want to do is shift this curve to the left. We want to be able to, with vaccination, generate an immune response which is specific to the virus, but do so without actually infecting the individual. And how do we do it? Yes, we can do it by mimicking an infection, by presenting to the immune system virus-like particles and triggering this multi-armed but time-taking process, so that when the real infection comes along, this adaptive immune system can kick in quickly and can kick in forcefully and protect you from not necessarily an infection, but from getting very sick and even dying. Okay, so ultimately what we want to do is prevent illness. Okay, so in order to understand how the vaccines work, we want to understand how the immune system works as well. So I will tell you about it. Again, this is a fairly complicated picture, and it's a picture of a very much simplified immune response. Things in reality are complicated, so bear with me. When you're first exposed to a virus, let's say this is SARS-CoV-2, and we call it an antigen, that virus infects the cells, it's replicated, but ultimately those viral particles are engulfed by a subpopulation of cells they're called antigen-presenting cells, which you'll see later on when we talk about the vaccines. And those are really important cells because they tell the body, I have not seen that shape before. I have not seen that molecular shape before. I think this could be a virus. We need to mount an immune response to it. And those antigen-presenting cells, which live in the tissues, recruit a traveling population of white blood cells. They're called helper T cells. And when they engage each other, when the match between what the presenting cells are showing to their environment and the helper cells are set to see on their surface, when they interact with each other, that activates the helper cell, which then go off into different tissues, and there's travel involved, from different parts of the body. And they go to different tissues and they trigger activation by cells that ultimately produce antibodies, as well as cells that ultimately specialize into recognizing cells that are infected with that same kind of an antigen and cause them to commit suicide. Okay, so this is how the adaptive immune system works. It's very specific to a particular kind of an infection that presents to the body a very specific molecular shape that then the immune system responds to. Okay, and it comes in these two branches that we already mentioned. One is the humoral or antibody, soluble antibody immune response, and the other one is carried out by cells that cause the infected cells to commit suicide. Okay, and the net outcome is both the formation of antibodies as well as preparation of cells that can kill infected cells, as well as the memory cells that remember next time when we see that virus, we will remember to do both of things all over again and do it much faster. Okay, now the thing to notice here is that this immune system is responding to patterns that it sees on the pathogen. And when it comes to SARS-CoV-2, the very first thing that our body sees is the viral outside. And that viral coat is made up of what I'm sure you've heard about is spike proteins. Those are the most visible, first visible molecular structures that are to which the immune system can see. And these spikes, these protein spikes, are encoded by a messenger RNA that is contained within the virus. And I want to point out that the coding for the spike protein is only a part of the entire messenger RNA, which is required in order for the virus to replicate itself, right? So it's just a fragment that encodes for the amino acid sequence that makes up for the protein that then makes up for the coat on the virus. And that is the first thing that your immune system will see. It's the first thing that the antigen presenting cell will recognize as being foreign. Is the thing that the antigen presenting cell will then present it on its surface, that then the helper T cell will see and respond to, and then show to both the eventual antibody producing cells, as well as the cytotoxic T cells. So that's the trigger that starts this entire adaptive immune system. And the trigger is the spike protein, which is encoded in a fragment of the viral genome, which is a messenger RNA. Okay, so I already mentioned that developing this adaptive immunity takes time. And here's the timeline as measured by what is called neutralizing antibodies, antibodies that are capable of recognizing a viral surface protein, binding to it, and then preventing the virus from infecting the cells. So when you first get exposed to a pathogen, say SARS-CoV-2, nothing much happens for the first 2-3 days visibly to you. But in the meanwhile, all that communication and travel is happening between the cells within your body, and the B cells start mounting their response, and they start producing and secreting the antibodies. So somewhere around 3 days, you start seeing the antibody secreted in your plasma, and as such, it can travel throughout the entire body and intercept viruses anywhere within the body. But then after a few days, without additional exposure to the virus, that those antibodies stop being elaborated. Basically not much is happening, so the cells stop that produce those antibodies stop replicating and kind of go into a hibernation. However, if you come back a couple weeks later, or 2, 3, 4, 5, 6, or many weeks later, and you're exposed to that very same molecular shape that's part of the same pathogen, those dormant cells, which have already learned how to mount an immune response that is specific, those memory cells, jump into action, they wake up, and they come up really, really quickly, numerically and quantitatively, and they start making large quantities of the neutralizing antibodies, so that should this be a real infection, they can stop it from spreading throughout the entire body and affecting all those organs that we were looking at later. And all that can be triggered by presentation of just these spikes that are normally present on a viral surface. So, how to build an immune response to a virus such as SARS-CoV-2 without actually infecting a person? There are many different ways of approaching it. One way of doing it is just inactivating the virus. Dr. Kieft was referring to inactivated viruses before. One way is heating it or chemically cross-linking it. But the whole point here is that to preserve the structure, that molecular shape that then the immune system can't see, and presenting it to the immune system so as to respond. Another way of doing it is just purifying that molecular shape, that protein, and injecting that. Another one is aggregating those proteins so that the body sees not just individual small proteins, but these aggregates that mimic the virus even more than the individual proteins and stimulate a more robust response. Another way of doing it is take a virus which has been gutted, take a virus which no longer can replicate inside the cell, but can infect those cells very efficiently, and replace its genome with that which ultimately directs the synthesis of these spike proteins as well. That's a really efficient way of getting cells to produce large quantities of immunity triggering proteins without actually causing any kind of a disease. It is also possible to just introduce into cells the DNA that ultimately can be transcribed and translated into these spike proteins, but that's in general rather inefficient process because our bodies very quickly degrade exogenous DNA. Same goes for RNA. It's possible to introduce just naked RNA, but the RNases and enzymes will degrade it really quickly, and it won't get a chance to be translated into the protein and trigger an immune system. So these nucleic acid vaccines really have to be protected in a way before so as to get into the cells efficiently. So this building an immune response without an infection, there are many, many different ways, each one with technical and medicinal pluses and minuses. And you can look at this slide later on if you're interested. There's a lot of pros and cons that go into it. So I won't go into the details here. I just want to tell you about the vaccines that you will have a chance of receiving soon, if not yet. And those are the RNA vaccines, the Moderna and the Pfizer, and the protein subunit vaccine Novavax, and the viral vector that just got approved, Johnson & Johnson vaccine that's made by Janssen. And I'll tell you how those work. Now, I do want to point out that the rest of the world has many different approaches as well, and those are quite efficient as well and saving lives already. So let's see how these vaccines work. All right, that's a really, really messy slide. Dr. Turner will call it a high yield note, too much information at once that what he's supposed to memorize. So I'll take you through this stepwise. in a stepwise manner and probably not cover absolutely every detail here. But before we do that, I want to point out, tell you about messenger RNA, right? So messenger RNA is that nucleic acid, which in cells like yours and mine, eukaryotic ones, are translated into protein. And so if the messenger RNA encodes the spike protein, then when that messenger RNA is present in a cytoplasm, regardless of its source, it will generate the spike protein. And that messenger RNA, in order to be recognized as something that's translatable into a protein, has to have certain structure. Otherwise, it's not translated, and in fact, it's degraded really, really quickly. It in between this it has this coding region that determines the amino acid sequence of, in our case, a spike protein. But to be seen as a translatable message, it has to be polyadenylated. It has to have this many adenosine tails, and it has to be capped properly. And also has to be capped properly. But in order to function as a proper vaccine molecule, has to have some very specific molecular characteristics. Again, we do not have time to go into it, but that was the big challenge for the formulators of the current messenger RNA vaccines. It's how do we make a message which is efficiently translated and has a sufficiently long lifetime itself in a cell so as to generate sufficient number of spike proteins, so which then can be presented efficiently on a surface of a cell and then trigger the immune system. So there are some molecular chemical modifications of the nucleic acids that stabilize the longevity of the protein, as well as enable the cells to still see this as a proper messenger RNA that is to be translated, and then a messenger RNA that is optimized for longevity and efficiency of translation. Best I understand right now, and there may be others who know better, a messenger RNA will live in a cell for maybe a couple, 3 days at its longest. That is a study that can be figured out, but I've not seen the study done. And during that time is going to direct synthesis of several thousand of protein molecules. And it's those protein molecules that ultimately trigger the immune response. Those are your actual tools of immunity. But without drowning you into too much detail, here we have our virus. And we're going to talk about how the Pfizer vaccine, which is a messenger RNA vaccine, works. It has this very high level of efficiency, and Dr. Turner will explain how that is calculated, and it is administered as about 1/3 of milliliters into your arm with 2 consecutive doses, two doses that are spread out over 2-3 weeks into your arm to give you the full immune response. But anyway, back to our virus, right? This is our coronavirus. It's got this large messenger RNA genome, and just a fraction of it encodes for the spike protein. So that spike protein gene was taken out by the Pfizer folks and chemically modified to increase its stability, and then encased into a protective lipid layer. Without this protection, it would be degraded very quickly, and it wouldn't be entering the cell very efficiently, and we need to get it into the cell to be translated. So this encapsulation was a tricky part as well, but they got it done very, very nicely, because now these segments of messenger RNA that encode only for the spike protein are taken up by the cell. The cells usually macrophages that are sitting around and patrolling your arm muscle cells. And so the messenger RNA gets in and it's translated into the protein as part of the cellular behavior. Those proteins also end up on the surface of those cells. There they are recognized as being something new, something that needs to be responded by. other set of resident cells. They called dendritic cells or antigen presenting cells, and they take up these spike proteins and say, this is something to alert the immune system to. So they present that antigen on the surface, on their own surface, and these marauding, patrolling helper T cells come by and say, oh, that is something that I recognize as well. We should get really upset about this and alert the rest of the body to it. So they get activated and they interact with these B cells in certain parts of your body. The B cells and the activated T cell interact, and that properly changes the behavior of the B cells so that now they're capable of secreting large quantities of these antibodies which are capable of identifying selectively and only particles that contain viral particles that show up as having the spiked protein. So by coating those viruses, the virus is unable to bind to the receptors in the body, so they neutralize, plus they're taken up by phagocytes and destroyed. Now, at the same time, as the helper T cells are activating the B cells in order to make plasma cells and large quantities of the secreted antibody, those same specialized helper T cells are activating that other arm of the immune system, the cellular immune system, where they're alerting a separate population of cells into recognizing infected cells and triggering them into killing themselves. It's killing the infected cells themselves, so they cannot serve as a breeding ground for additional viruses. So all this is happening, all this is taking time, usually a couple of weeks, but at the end what you end up with, yes, this immune response, cellular immune response, as well as the humoral immune response, as well as these memory cells that are capable of making more antibody and more targeted T cells upon additional exposure, so that everything can happen again and again upon repeated exposure to the same antigen. So given this pattern, a vaccinated person with the Pfizer vaccine is considered to be protected about 2 weeks after the second exposure, right? So, and it takes about 3-4 weeks between the first exposure and the second exposure, and then 2 more weeks. But after that, you're considered well protected in that you're not likely to, your protection will not permit for replication of viruses in your body to the extent of making you sick enough to be hospitalized or even die. So that's the two shots that you would get with the Pfizer vaccine. And here's the data that Dr. Turner is going to dwell upon much more thoroughly. This is really a spectacular feat by humanity. The results are tremendous. While the placebo group kept getting ill and hospitalized and even dying, the vaccinated group was protected and has showed the potential of downgrading an infection from a deadly event to that of an experience with just a normal virus that takes a couple of weeks to get over. So that's really the goal here. And this is the data that Dr. Turner will focus on, but it's really, really spectacular. Out of 40,000 volunteers split between placebo and control, the placebo folks got were about 170 cases of severe infection, whereas there were just a few 8 cases among the vaccinated group. Just a tremendous difference. Okay, and I know this is going on and on and on, but I just want to tell you about the other vaccines. The Moderna vaccine is essentially the same. There are slight differences in which piece of messenger RNA is used to encode the spike protein, slightly different chemical modifications on the messenger RNA, slightly different coating of the messenger RNA containing viral particles, slightly different quantity of RNA that is encapsulated and then administered, but the process is exactly the same. Those viral particles are taken up, protein is translated, it's presented to various branches of the immune system, and upon repeat of exposure ends up giving you this both immediate as well as the long-term memory response to the spike protein and thus to the SARS-CoV-2 vaccination. The Johnson & Johnson vaccine that just got approved, and it's looking really funky here, is a little bit different. And it's different in the sense that, if you can see properly, is different in the sense that the messenger RNA still gets delivered, but it gets delivered via an indirect route. The messenger RNA was converted into double-stranded DNA, which a gutted adenovirus can deliver very efficiently. And because instead of delivering the messenger RNA that can be translated only a few times, this DNA can be transcribed many, many times over a period of potentially a week or weeks, so as to generate many more copies of messenger RNA, which can continuously get translated and continuously trigger this immune response. And as such, all you need is really a single copy, a single injection of this viral adenovirus and still receive a long-term response. Let's see, I don't know what happened to this slide, but let's see, hopefully the other one's going to be better. Okay, so the Johnson & Johnson vaccine, instead of introducing messenger RNA into the cell, introduces into your cell via a non-replicating virus DNA that has a longer lifetime and can generate many more copies of messenger RNA and thus many more copies of the protein. Now, the Novavax vaccine is again different in that the spike protein itself is synthesized directly in insect cells, and then it's purified and combined with what we call an adjuvant, one that helps to trigger a really strong immune response. And it's these spike protein particles along with adjuvant that are taken up directly by the dendritic cells and then presented on the surface and then exposed to an interaction with the helper T cells that then trigger the two different arms of the immune response and ultimately give you both the short-term and the long-term memory. And again, here you are protected 2 weeks after your second booster. And the efficiency of the Novavax vaccine at this point is about the same as that of the messenger RNA Pfizer and Moderna vaccine. Now, before I turn it over, I just want to tell you what's in the Pfizer and Moderna vaccine ingredients. Those are the ones that we've been seeing already in New Mexico. We'll probably be seeing it for a while longer. So here are the ingredients. Both Pfizer and Moderna have messenger RNA. They're slightly different flavors, slightly different modifications, but basically they encode for the same spike proteins. The lipids are the ones that protect the messenger RNA and help it be delivered with greater efficiency into the cells. And both those lipids are described with very fancy names, but basically those are really just the protein, similar kind of lipids as our own cells are made up of. There's slight differences between the two, nothing very dramatic. And then the rest of it is just salts that balance the pH, the acidity of the injection with, and make it compatible with your body tissues, and some sugar that make those viruses stable upon freezing. And you want to be freezing the vaccines so as to protect the generally fairly labile messenger RNA, right? So one thing feeds into the other, and a balance needs to be found. But all in all, these are really simple ingredients. There's nothing dramatic. The only thing that potentially is new to some people's immune system is this polyethylene glycol. It's a polymer that's actually present in many foods and shampoos and everything in between, but it is thought to be the one component that could possibly be responsible for these very strong immediate immune response says by just a handful of individuals who've been exposed to this vaccine and cause anaphylaxis. Now, even that anaphylaxis is super rare. It's been observed somewhere on the order of 5 in a million of vaccine recipients, the SARS-CoV-2 vaccine recipients. And that is 1,000 times rarer than anaphylaxis, which is experienced by your regular flu vaccine recipients, right? So even that has been brought down. Here it is, okay? This is what we try to do with the vaccination. We're trying to generate a specific adaptive immune response that precedes your exposure to the virus so that you can respond to the virus and inactivate the virus before it has a chance of infecting your cells and tissues. And the way we do it is by getting vaccinated. It is true, as many others will tell you, is that we need to continue to be observe the social distancing and wear masks, because we can still be infected and can still spread the viral infection, but we will not get as sick as we would be otherwise, nor would we be infecting others with the heavy viral load that could get them really, really sick. So we're pushing with vaccination. We are preparing for the subsequent onslaught of the real virus so that we can respond to it very, very quickly. And we're doing really well, as Dr. Kieft told you. In New Mexico, we've got some of the best, fastest vaccinations in the country. So this is really exciting. And Dr. Turner will tell you what are the post-vaccination side effects and what is the efficacy. So go out and get vaccinated. And yes, I am finally done.
Thank you, Dr. Rogelj. That was so great, really informative, really helpful. I have listened to this twice and I learned even more the second time. So that was super helpful.
Thank you for your patience.
So we've been getting a couple really great questions in the Q&A, but we also got some really good questions ahead of time. And so I was hoping that I could ask some of them now. And anyone, feel free to give a response. I think these are questions that would apply to this section, but any of our panelists, feel free to answer. So I think one of the things that students will be particularly concerned about is if they've already had COVID, do they need to get vaccinated? And how does the vaccine help you, or does it, once you've already had COVID?
I can answer that if you like, unless somebody else wants to pipe in. So let me get started. So once you've had COVID, indeed, you will have some sort of an immune response, but it depends on how on the degree of the initial infection, right? So how your immune response worked out after your viral infection. And some people will have very strong response and some people will have a very mild response. And of course, then we have the asymptomatic infection. And it is entirely possible, both with the asymptomatic infection as well as with the very strong response to the infection, that your immune response isn't very strong. With the asymptomatic infection, it's possible that your immune response just wasn't responding very strongly, so you're really not feeling the symptoms, which very often are the result of your immune system gearing up. And those who had a very strong, became very ill with the infection, those people may be actually the ones that have a very weakened immune response as well. So if nothing else, a booster would serve them well. And so universally, regardless of the vaccine that's available, the doctors are, and those in the much better know than I am, are recommending absolutely get vaccinated. Do make sure that that sufficient amount of time has passed since from your recovery from the actual infection. They're recommending about 60 days post-recovery, at least 60 days, but definitely get not just get vaccinated and get full 2 doses if you're getting the vaccine that requires 2 full doses. And I might as well mention here is that they're recommending that for women who are pregnant. You just have to watch out for the fever that could be elicited by the vaccination, as well as women who were pregnant when they had their natural COVID infection. They are still telling yes, absolutely, you need to get vaccinated. And that has to do with boosting up the immune system, as well as having some protection against any sort of variants that might be emerging. So I think the answer is pretty unified. Absolutely, you should get vaccinated.
Great, thank you.
I'm just going to
Dr.
add,
Chakerian, could you add some?
Yeah, sure. Another way of thinking about this is that, when you're infected with the virus, viruses have developed all sorts of different mechanisms to prevent immune responses against them. They have ways of sort of turning off immune responses. And so, that's one of the reasons why for some people, immune responses against this virus are not particularly strong. Now, the whole point of vaccines are to maximize immune responses against a target. Okay. And so, when people have looked at the immune responses to the vaccines compared to people who have recovered from viral infection, the vaccines generally elicit stronger immune responses than natural infection does. And so, getting the vaccine is actually going to protect you better than if you recovered from COVID infection. That said, most people who are recovered will probably be immune for a while, but they are recommending that people who have recovered get vaccinated. And right now, that recommendation is to get multiple vaccine doses if it's a 2-dose vaccine. But there's emerging evidence that probably even just a single dose of vaccine is going to be sufficient to give you strong responses. So it could be that those recommendations will change at some point. That would also help save vaccine for the rest of us who haven't been infected. So it's a great question, and it's more data to sort of support the reason to get a vaccine.
Yeah. This is a question that I've been having about, and it has to do with the immune response and the strength of the immune response. We saw earlier that most of the cases that result in severe illness and death are biased towards elderly people. Is that in part due to a reduced immune response in elderly people, or is it really the comorbidities there? And then sort of relatedly, does that have, I've heard anecdotes that younger people have stronger symptoms after getting the vaccine than older people do. And so can anybody shed light on whether those are connected at all, whether that's sort of anecdotal and not real or anything like that?
I could jump in on that one too. So there seems to be evidence that I think someone's asking for to stop screen sharing right now so that we can do think it'd be
panel
great.
view.
Thank you.
So I think there's evidence for both of the things that you mentioned. So there's evidence that suggests that the more quickly you start making immune responses to a COVID infection, the faster you're going to recover and the less your symptoms are going to be. And generally, younger people make immune responses faster than older people. It's sort of a rule of thumb. And there's various cells that can help induce stronger and faster immune responses, and they tend to be more prevalent in younger people. So I'm sure that is part of it. And then the second thing is what you said, the higher levels of comorbidities in older people who have heart disease, who have diabetes. And of course, that also is going to affect both immune function, but also the consequences of getting infected.
Thank you. Dr. Rogelj, did you have anything to add there? Or?
Oh, I got distracted by the questions in the chat here.
That's okay.
Did
No
you
problem.
want to address any of those? Or an
Um,
interesting thing about formaldehyde.
Oh yeah, if you could, yeah. So we have a question from that says something the anti-vaxxers consistently bring up is the vaccines have formaldehyde. Is this true for shelf-stable purposes? Is this true of the COVID vaccines? And why are they not included? Or what's the purpose of the formaldehyde, and where is it? Are these vaccines different in any way?
Perhaps I can get started on that, and then
Yeah.
Dr. Chakerian, you can add to it. But formaldehyde is sometimes used in order to create vaccines. Formaldehyde is a denaturant, and it preserves it sort of kills the virus in an analogous case, kills the pathogen, but at the same time preserves the shape of the proteins. And for the most part, it's removed thereafter once all the proteins are preserved and prepared for making of the actual injectable vaccine. But the messenger RNA, the viruses that we're dealing with now do not contain any formaldehyde. In fact, they have to be kept far away from formaldehyde because a fixed messenger RNA just wouldn't work very well, right? So the preservative, there is no formaldehyde in the vaccines that have been approved that I know of. They do not need fixing because they don't need killing, and they mustn't be fixed because they require to continue functioning as a natural molecule would. The preservative in the case of the vaccines is cold. That's why they need to be frozen or and protected in their lipid membranes. But that is not something to be concerned. Exposure to formaldehyde is absolutely not something to be concerned about with the Pfizer, Moderna, and Johnson & Johnson and Novavax.
Awesome. Thank you. That's really helpful.
Can I weigh in here for a second too before
Please, Dr. Kieft, go ahead.
Comes in and gives us the definitive word. Another reason that formaldehyde is used is to not only preserve but slightly modify proteins, so that's used for a tetanus toxin, diphtheria toxin, and as Snezna said, it's removed afterwards. But some of that formaldehyde modifies the structure of the toxin, and our immune system then protects us against the actual toxin because of this toxoid, the modified toxin.
Okay, that makes sense. Thank you. All right, any other points? I think we can, this sort of discussion about ingredients and stuff is a good transition point for Dr. Chakerian to come in and tell us about how the FDA approval process works and what goes into that. So Dr. Chakerian, would you like to take over?
Sure, sounds great, thanks. So everyone can see that?
Yes.
Okay, so I've gotten this question a lot. How did we get this vaccine so fast? And I remember at the beginning of this pandemic, there was a statistic that was cited that the fastest that we've ever moved in developing a vaccine was the mumps vaccine back in the 1960s, which took about 4 years to develop. And here we have a vaccine that was developed less than a year after this pathogen was identified. It's really quite phenomenal, but I think it also raises a lot of really legitimate questions about the process for approving vaccines. And so I'm going to talk a little bit about that in just a few minutes. So, okay, so this is a typical vaccine timetable. And you can see that if we had followed this typical vaccine timetable, we wouldn't have a vaccine, for about 13 years, which I think would be really depressing. And there's a lot of different steps along this. There's often academic research, and here it has that as 2 years. But I'll tell you from experience that the academic research part can often take much longer than that. And that's, trying to figure out, like, what is the best vaccine strategy? What parts of the pathogen do we want to target? These are important questions, and, the answers to those questions really depend on the pathogen. And then often after that stage is a preclinical stage where, those vaccines are then tested, usually in animal models. Does a vaccine protect a mouse from infection by the pathogen? And does it infect or protect a monkey? And those can often take several years. And then only after a vaccine is able to pass these sort of early preclinical steps does it actually go into people. And I'm sure many of you have heard about these different phases of clinical trials, but they're sort of basically broken up into phase 1 clinical trials, where there are questions like, is the vaccine safe in a small number of people? Do we try different doses of the vaccine, and what sort of immune responses do we get at those different doses? And are those different doses safe? And then once you pass that phase 1 sort of, the phase 1 trial sort of part, then you can move on to a larger group of people, and that's a phase 2 trial. And there you're asking a question again, when we expand the number of people that we're testing this vaccine, is it still safe? Does it still induce the sorts of immune responses that you'd like the vaccine to induce? And then, and only after it passes phase 2, does it move into phase 3 trials. And phase 3 trials are typically really large numbers of people. And it's really asking a very specific question. Does the vaccine actually work? Does it protect from disease in that large group of people? And you've probably seen some of the large numbers of people that the current COVID vaccines have been tested in. So for example, Moderna and Pfizer were tested in 40,000 people. So really, really large numbers of people. And that also allows you to generate safety data. When you test it in a lot of people, then you can start to try to find very rare side effects that those vaccines might cause. And then usually what happens is after phase 3, when you see if a vaccine works or not, that's when the sort of scale up begins. That's when you start modifying factories so that they can actually manufacture the vaccine. And you actually start making the vaccine and seeing if you can make that vaccine at scale. So those are also really important questions in moving a vaccine from early clinical study into the real world. And then there's paperwork, the approval process, where the company or the group that's making the vaccine submits lots and lots of documentation in the United States to the Food and Drug Administration, and the vaccine it's decided whether the vaccine works. And then there's the distribution. And that also sometimes presents some logistical problems, not necessarily in the United States, but often in the world that can be a real issue. So again, this process can take a really long time. So here's what we've done so far with COVID. And we started this again in early 2020, and we had our first approved vaccine in December 2021. And all of those steps that I mentioned before that are done typically with the vaccine were also done with the COVID vaccine. But you can see that a lot of these steps overlapped. So for example, the initial studies that were done to test whether these vaccines worked in animals were done simultaneously with the early studies in humans to see whether these vaccines were safe and induced immune responses. Phase 2 trials overlapped with phase 1, phase 3 overlapped with phase 2. And during that period, there was also efforts to try to secure the manufacturing capabilities that would allow this vaccine to be produced and the, ultimately billions of doses of these vaccines to be made. So again, none of these steps were omitted in the COVID vaccine process. They were just accelerated. So again, how do we move so fast with this virus? And again, I think that's a really good question. And I think there are a few answers to that question. And the first is that, we actually know a lot about coronaviruses. There are coronaviruses that normally infect humans. There are several coronaviruses that actually cause essentially the common cold, although not all colds are caused by coronaviruses. And so knowing a little bit about the biology of these viruses was really helpful in trying to, again, decide what parts of the virus to target and what sort of vaccine strategies to use. We just heard a little bit about the vaccine technologies that are being used to develop these vaccines. I think it's really important to point out that these are really new and cutting edge vaccine technologies that can be implemented very, very quickly. Particularly the mRNA vaccines like the Pfizer vaccine and the Moderna vaccine. A third really important point, and this maybe is the most important thing of all, is that there have been huge investments in vaccine development. The federal government has spent billions of dollars in pushing vaccines, helping companies. Companies have also made huge investments in these vaccines. And those sorts of investments have also allowed all these things to happen. When companies know that they're going to be able to get vaccine out into people, then they're more willing to change the factories so that these vaccines can be developed even before the vaccines have actually been produced. And then I think another really important point is that one of the critical steps for the approval of a vaccine is showing that it works. And maybe this is a little bit of an unfortunate problem with the COVID pandemic, but because so many people were getting infected with this virus while these vaccine trials were going on, those trials were actually able to get data much, much faster. So when you see people in the placebo groups coming down with the virus, the more people in the placebo group get the virus, the more quickly they're able to get data that shows whether the vaccine works or doesn't. So the magnitude of the pandemic has also played a role in getting us these vaccines quicker. So I'm just going to, in a couple of slides, I'm just going to talk a little bit about some of these points. We know a lot about coronaviruses because there have been cold-causing coronaviruses, but there was also SARS-1, which was an infection that emerged in China in 2003 and causes a very, very severe disease. And there were a number of efforts to try to develop vaccines against SARS-1. But fortunately, this virus actually disappeared. And we didn't need to use those vaccines. But the sorts of technologies that we used to try to make potential SARS-1 vaccines were then used for SARS-2. And then in 2013, there was another coronavirus called MERS, which is actually transmitted by camels and was an infection in the Middle East. And again, this virus from 2013 was also another impetus for trying to develop vaccines against coronavirus. This is also a very serious virus that causes a high mortality rate. The advances of vaccine technology, we just heard about that, these mRNA vaccines and DNA vaccines and adenovirus vaccines. So there are really no FDA approved vaccines that use these technologies, although some viral vector-based vaccines like the adenovirus vaccines are actually in clinical use, including a Ebola virus vaccine. But these vaccines have been in development for years now. And for example, Moderna has been developing RNA vaccines for a whole variety of different pathogens, for Zika virus, for cytomegalovirus, for influenza. And many of those RNA vaccines have actually gone into humans in clinical trials. And so the technology that they're using to make these coronavirus vaccines has been used before, and it's shown to be safe in these early phase clinical trials with other viruses. So even though these technologies are new, they're not new in the world of biotechnology. So that has also helped, move these vaccines quickly into people. So again, this is just sort of a slide showing many of the leading SARS-CoV-2 vaccines. And we just heard about the RNA vaccines that are approved and their efficacy is quite good. The Johnson & Johnson vaccine that was just approved last week also has quite high efficacy. It's not quite as high as the RNA vaccines, but it seems to be effective after one immunization. So that's an advantage. We're going to hear more soon about the AstraZeneca or Oxford vaccine, which is also an adenovirus vaccine. I think that the best estimate for when this vaccine will be approved is either late March or early April 2021. So we very soon could have a fourth vaccine at our disposal quite soon. The Novavax vaccine this is a protein-based vaccine, is also in late-stage clinical trials, and it's possible that that might follow the AstraZeneca vaccine quite quickly afterwards. And then we have these more advanced vaccines that use even more potent vaccine technology. Here's one that I'm not going to go into, but this is my favorite vaccine that's in clinical trials right now. It was actually developed at the University of Washington, which is my alma mater, and is now being commercialized by a company in South Korea. So again, we have all these sorts of technologies that are rapidly moving into people. And one of the great things about many of these technologies is that people are worried about different variants of the coronavirus. And these vaccine technologies are quite flexible. The genes that these vaccines encode can be swapped for the new variants. And Moderna and Pfizer, and I believe also Johnson & Johnson, are already developing next generation vaccines that incorporate these different viral variants of SARS-CoV-2, which may or may not end up being a problem. So again, that's, I think, a good thing to think about for the future, that these vaccines will potentially be able to keep up with the virus as it evolves. So I think that's all I have. I'll stop sharing.
Thank you, Dr. Chakerian. That was really informative. It sort of seems like a war effort in how much investment has been put into this year. And honestly, it feels like we've been fighting a war in our own houses of just trying to not get coronavirus. Do you think that this is going to be the model going forward, is like sort of ramp up production and compress this timeline? Or is it going to revert back to the long-term sort of getting vaccines ready over decades?
Yeah, I think your analogy to the space race is really a perfect one. It just goes to show, when we put our minds to something, the technology is advanced that we can do things very, very quickly. But a lot of this has to do with just sort of the willingness to do that. And obviously with this pandemic, because of the severity of the problem, the fact that so many people have died, that was really impetus to do this quickly. I think with other vaccines, there's the potential to do something similar, but maybe there isn't the urgency that there has been with this virus. So, and I have to say that, the fact that so many people were being infected and allowed those clinical trials to reach their endpoints really quickly made a big difference in approving these vaccines. I worked on a virus called human papillomavirus, which causes cervical cancer many, many years ago. And one of the problems with that virus is it takes decades from you to get infected with HPV to actually develop cervical cancer. And so that becomes a bit of an issue when you're trying to show that the vaccine actually prevents cancer. So, it depends on the particular pathogen, how quickly you can move. But sure, I think this shows that we can, if we set our minds to something, we can do things very, very quickly, which is great.
Yeah. I mean, it's like incredible, just the tech, like being able to transition from basic understanding of coronavirus biology, from RNA sequencing to all the way to, okay, we're going to design a specific RNA to this genome, and it's going to induce an immune response. That's just fascinating biotechnology. That is, I mean, we're lucky that we live now, right? That if this would have happened 20 years ago, it would have been devastating. Like with if SARS would have been like as infective and as transmissible as COVID, I think we'd be in a world of hurt. So I think we're lucky from that perspective. Okay, we have one question. I think Dr. Turner is typing live, but I'd also like to transition over to her presentation anyway. So maybe Dr. Turner, instead of typing, you can answer it live if you don't mind. And this question is, what was the selection process for participants in the efficacy trials? Because you're going to talk about efficacy in just a second.
Right. So I didn't find very much information on how participants were chosen. Everything that I kept reading was, it was a certain age group between either 16 for one of the vaccines or 18 for the other, through, adulthood. And then one of the vaccines, it's Novavax. And yeah, Dr. Chakerian, you may know more details on this. Mainly it was healthy adults, but for the Novavax, they did allow some adults to be participants in the trial as long as their underlying conditions were well controlled. But for the other trials, I could just find essentially healthy adults. But yeah, Dr. Chakerian, you can jump in.
Yeah, so I mean, I think the goal for phase 3 clinical trials was to really enroll a cross section of people in society. Mm-hmm. So for example, in the Johnson & Johnson trial, you saw people who had diabetes, you saw people who had other comorbidities. And really the goal there was to see how well this vaccine worked in all sorts of people, in old people, in young people, in people of different ethnic backgrounds. That was a really important component of the trial so that we could really see the breadth of efficacy in these vaccines. I have to say that I volunteered for one of the vaccine trials for the AstraZeneca vaccine, and I received 1 dose of that vaccine, and then I actually got the real vaccine. So I jumped out of the trial. But at the time, they were taking all comers, and then right after I enrolled in the trial, they restricted it to people who were over 65 who had pre-existing conditions because they needed more people in those groups to test the vaccine. So I think that's a really great component of these trials, the fact that they were testing it in such a cross-section of our society.
Great, thank you both. All right, Dr. Turner, why don't you give us a guide to safety and efficacy of these vaccines?
I can do that. And I know we're about at time, so I will try and go quickly. But I just wanted to start out by grounding us a little bit. We're all familiar with the flu. And one of the biggest fallacies I kept hearing in the beginning and some now is that, COVID is just like the flu. It's not that much worse. And so here are the flu deaths in the United States over the years. And then you compare that, as was noted earlier, over 500,000 people have died in the United States alone from the coronavirus, SARS-CoV-2. So we all know now it is a big deal. It's not the flu, it's much worse. And that's why these vaccines are so critical to us moving on and to staying safe and to keeping those around us safe. And so again, I just wanted to point out a few different vaccines that we have historically. So the flu vaccine we're all familiar with. Many of us take it every year. There are lots of flu vaccine campaigns, even here on campus. We're comfortable with it. It's great. And it's about 40 to 60% effective. And we're okay with that every year. So getting these mRNA vaccines that are, 95% effective is really incredible. And I want to point something else out. As Dr. Kieft mentioned earlier, just how wonderfully impactful the measles, mumps, and rubella vaccine is. The reason it's so impactful and the reason it's really efficacious at 97%, probably one of the best vaccines we have so far, is that it's a live attenuated vaccine. So what that means is you've essentially got the virus still alive. You're introducing it to your body and your body's saying, oh my gosh, there's a live virus. Let's kick off every pathway of the immune system, which is what Dr. Rogelj really beautifully explained. And so the flu vaccine is an inactivated virus. It's dead. It's what we talked about earlier. So that's why the efficacy is a little bit less. What's so beautiful about these mRNA vaccines is that it's not a whole vaccine. It's not killed. It's not attenuated. It's just that one spike protein that kicks off every aspect of your immune system really beautifully. And it's, 95% effective, which is absolutely incredible. 2 doses, of course, are required. And then we've got the Johnson & Johnson vaccine, which is also really impressive at 72% effective here in the US with just one dose. So I think it's really amazing, the feats that we've overcome. And we've got these really safe vaccines. I know people are worried about them being able to integrate into your DNA. No, mRNA is in a completely different compartment, and it's translated and it gets kicked out as that one little protein that's presented to your antigen presenting cells, as Dr. Rogelj explained. So it's not going to change your DNA, but it's still effective with that one little spike protein. So I had this on last week, and this is before the Johnson & Johnson vaccine was approved in the US, just comparing Pfizer and Moderna. And we now know pretty much that they both require 2 doses. They're both about 95% effective. And as was mentioned earlier, this is definitely considered a high yield slide if you want a lot of information in a small space. So it's something you can screen capture, or I think the slides are available later, but it just shows, after 2 doses, you get really well coverage, but even after 1, you get about 89 to 80% effectiveness, depending on which Moderna or Pfizer vaccine you get. And so I was curious, as were others, about how efficacy is calculated. How do we get that 95%? So I'm just going to quickly explain here using the Pfizer data from their clinical trials. They enrolled 43,660 subjects, and these participants were split evenly into about the number here, 21,830 subjects per group, so placebo or vaccine. And over time, they watched the number of people who became infected with COVID, who received the placebo, which is 162 people, and only 8 people became infected with SARS-CoV-2 upon receiving the Pfizer vaccine. So from that, we calculate the risk of infection, which was 0.74% in the placebo group and 0.04% in the vaccinated group. So what does that mean? How do we scale this up? So what we do is we take the population of people in the United States, which is about 328 million, and we multiply the infection risk, that 0.74% by population, and we can see about 2.5 million people are theoretically expected or have a risk of becoming infected without the vaccine, whereas only 131,000 people are likely to become infected with SARS-CoV-2 once they receive the Pfizer vaccine. So again, how does this translate? What does this mean? In order to derive our 95% effectiveness, we take the difference between the risks in both groups. So you have a 0.74% risk of becoming infected with SARS-CoV-2 when you do not get a vaccine, and a 0.04% when you do. So that's a 0.7% difference in risk of infection. And I was underwhelmed when I first saw 0.7%, like, what is this? This is a tiny number. But it turns out small percentage differences, once we scale up to population, really do result in huge numbers of people having a less likelihood of becoming infected with SARS-CoV-2 upon receiving the Pfizer vaccine, or Moderna, or any of the others for that matter. So to get the 95%, we take that difference in infection risk and divide it by the percent risk that you would get SARS-CoV-2 if you didn't get vaccinated, and that's where we get a 95%. So under the same conditions as the study, the vaccine reduces the risk of infection by 95%. And as we know, that's huge and exciting and really important to keep in mind. So you get the vaccine. What happens afterwards? Well, with Pfizer, again, we've got that number of clinical trial participants, 16 years and older. Most common side effects are what you would probably what you've heard a lot about: injection site pain, and this, I should mention, is after the 2nd dose. So injection site pain, fatigue, headache, muscle pain, chills, joint pain, and fever. And this is your immune system responding and sending off all those signals. It's not a viral infection. You did not get COVID, even though you may feel pretty lousy after the 2nd dose, or you may not. Moderna, same thing. It's essentially the same commonly reported side effects, and they had 30,350 clinical trial participants, 18 years and older. And then we come to J&J, who had 45,000 participants. And so I just put this here so you can see everything side by side. Again, the Pfizer and Moderna side effects were reported after the second dose. And just to go back to what Dr. Rogelj beautifully explained, the reason that the side effects are worse after your second dose of these 2 mRNA vaccines is because your first dose served as a primer. You've got some immune response, but once your body has built up, a memory to the spike protein, it's going to respond really vigorously, and it turns on everything, and they send out signals to each other, and your cells are talking to each other saying, let's get rid of this protein. And so it's your body sending out those chemical signals that makes you feel that injection site pain or get the fever, which is reported. And just speaking from experience, I was vaccinated about a month and a half ago with Moderna. My arm hurt after the first shot. After the second shot, I want to tell myself I have a super immune system because I got a really high fever and I felt lousy for about 3 days. But I know people who received the same vaccine and they felt tired for maybe a day and they were fine. So it really varies, but I'd rather feel lousy for a couple of days knowing that I received a vaccine that's going to protect me and I won't have any long-term effects versus taking the risk of COVID, feeling lousy for weeks or months, and then not knowing how my body's going to respond in the future. And so this, again, was just focused on the Pfizer and Moderna vaccines last week. And it essentially sums up what's going to happen. So you get your first shot and you may feel tired after a couple of days. And then for 21 days later for Pfizer, you get your second shot, and 28 days later for Moderna. And that's when you really start feeling, or not, the symptoms, and you are then 95% protected. And I think it's really worthwhile. And these vaccines, as you now know, are safe. And now how your body's responding, which I think is really beautiful. And I'm really glad Dr. Rogelj went into all of those details because it's really magical how coordinated your body is to protect you. And I believe that is all I had. So I can stop sharing now.
Thank you, Dr. Turner. That was really great. Really nice to see that it's safe, effective. Yeah, take a few days off. Plan to take a few days off after you get it. Maybe get it on a Friday if you can.
That's what I did.
You can take yeah, perfect. Cool. So, and yeah, we had some questions about the symptoms, about how that happens. And I think you did a nice job about explaining what those symptoms were. We had some additional questions about has there been any cases that we know of death or debilitation or anaphylaxis or anything like that? What are the most severe responses and how common are those?
I wish I had the numbers. I think Dr. Rogelj, in your last presentation, you had the anaphylaxis numbers, but it's very rare. I think it was like 5 out of
a million.
How many? Yeah, a million.
A million, yeah.
And again, that happens within, they make you wait 15 minutes after you get your vaccine. It's really rapid. It can happen 5 to 30 minutes later. It's not something that you'll get later on to where you're left alone. And it's really rare. And death, no. There's no reported deaths directly linked to the vaccines.
Which is incredible considering how many deaths have happened since the vaccines have been started introducing. None among the vaccinated people, right? So at least due to the vaccine. I'm not sure if anybody has died of COVID despite being vaccinated. I'm not aware of any, but I'm not educated on those numbers.
There was there was an individual who died some hours after being vaccinated, and some trials were stopped, right? Maybe one of you knows better, Dr. Chakerian, but they looked into it and it was completely unrelated to the vaccination. And the anaphylaxis is almost predictable. So they warn people, have you ever, had an acute reaction like this, and be extra monitored. But I have not heard of vaccine actually triggering a death.
Those people who go into anaphylactic shock, that 1 in a million, they'll be injected with epinephrine immediately, which restores
Yeah.
homeostasis. They will be okay.
And that's in contrast. I was just asked to review some of the Johnson & Johnson data for the state. And in the Johnson & Johnson trial, there were 8 people who were in the placebo group who died of COVID infection. And of course, none of the people who got the vaccine died. So again, it's always a risk assessment. The vaccines are very, very safe. COVID is not safe.
Right.
Yep. Thank you. And anaphylaxis is the primary, only the serious concern about it, even given its rarity. That's the only major outcome that's been, other than these minor side effects like fever and chills and stuff.
Yeah.
Is that correct?
Absolutely, to the best of my knowledge, and I've read lots of papers.
Yes.
Okay, great. All right, so the final bit I want to close on is a little bit, a lot of the students were concerned about, and I don't know if any of us have any answers, but I want to ask it just to see if any of us can crowdsource some information. Is how soon do we think that students are going to be able to be vaccinated? And is there going to be, like, in terms of people in their 20s, in their teens and 20s, is there any sort of order to that? So one of the students posted a question that they have the sickle cell trait. Can they get priority for getting the vaccine? And finally, is there any strategy in terms of like students living in group situations, like in the dormitories? Can they get maybe prioritized because of the likelihood of spread? Or is that not being incorporated into the vaccination strategy? I don't know if we have any answers to this. I don't know the answers, but I just wanted to raise those questions and see if anybody did.
I could take some guesses if you like. So to my best understanding right now, the hope is that by the end of May, the vaccine, any one of the flavors, will be available to everybody, to all adults in the United States. Now, how quickly everybody will be vaccinated is a separate question, but it could be just a matter of weeks thereafter, assuming that people who want the vaccine will have access to it. So we're looking at a really quick turnaround time. I did look up the question about sickle cell disease for the carrier, and there is a link that I found that basically says absolutely that person should get vaccinated. Not clear about the priority, but there are no anticipated negative consequences as a result of their either carrying the trait or actually having sickle cell anemia. Then there was another question about priorities.
Yes, before we move on to the next question, I just wanted to add to that is all of the students should get registered and incorporate any pre-existing conditions like that onto your registration profile on the New Mexico Department of Health, because they will, if those traits like sickle cell or diabetes or asthma do increase your priority, the New Mexico Department of Health will incorporate that into your decision.
And I think that sickle cell trait actually does qualify as something that would put you higher in the list. So I would definitely add that to your profile.
Absolutely, yeah.
Agreed. Great. And then the last question was, is there any strategy in this population that's sort of highly likely to spread the virus about that? I'm not aware that that decision has been made at all, but I could imagine maybe a more effective vaccine strategy would be to target it towards group living situations rather than students living on campus versus students living off campus. Are there any plans underway for that at all?
I do not know of any, but I imagine if there will be plans made, they have to be made really quickly here, because the vaccines will be becoming available. I would recommend that everybody continues to practice all the safety measures until well past middle of December.
I think right now, the state's main priority is sort of a logistical one to try to get the vaccine into people as quickly as possible and as logistically feasible. But I think at some point, as more vaccine becomes available, then we might start to see these sorts of questions get answered. So for example, the Johnson & Johnson vaccine, which is effective, but there are some questions about its efficacy in people who have comorbidities. And so this might be a great vaccine to give to young people who will respond very well to the vaccine and are at lower risks of severe disease as it is, and maybe not as good a vaccine to give to people who are, elderly. So, right now that's not what the state is doing, but at some point that might be part of the equation. So we'll just have to see how this evolves as more vaccine becomes available.
Yeah, good point. And you mentioned earlier that the Johnson & Johnson had a really diverse trial. Did they break that down by age class of the efficacy? I knew they broke it down by country, where it was 72% effective in the US, but lower efficacy in like South Africa.
Mm-hmm.
Did they break it down by age class, where there was higher efficacy in younger people for the Johnson vaccine?
Yeah, and I would say the differences are a little bit subtle. And as you start breaking it down into smaller and smaller groups, then the error bars get bigger and bigger, the confidence intervals get bigger. But there did seem to be some trends suggesting that younger people responded better to the vaccine, or at least efficacy was a little bit higher in younger people. And there were maybe I shouldn't talk about this now, but there were a few concerning things about efficacy in, for example, people with diabetes. Where efficacy was much lower. And again, that could be a statistical quirk, and it's still a good vaccine for most people. But if you have diabetes, you might want to ask for one of the other vaccines. For everybody else, it's probably fine. So.
Great. That's super helpful, I think, information.
And I should say that those differences were not as apparent with the Pfizer and Moderna vaccines. Those vaccines worked extremely well in all age groups and all demographic groups.
Cool. Good news. Okay, well, I think that's a great place to conclude. Is when your turn comes up, please go get vaccinated. It's going to help you, and it's going to help the tech community. It's going to help the New Mexico community. It's going to help the United States. It's going to help the world. So please get vaccinated, and feel free to share this information with the people that you know. I've made this available it's live streamed on YouTube. I'll send out the link to the YouTube link once it's fully processed by YouTube and everything. And I'll make the slides available from all of our speakers who so graciously joined us and donated their time and intellect to this effort. I really appreciate it. Thank you, participants, for joining and asking such great questions. Students, you continue to impress me. I'm very new to New Mexico Tech, but I am just like really thrown by how thoughtful and dedicated you students are. So thank you very much. And most importantly, to our panelists, thank you for your time. Thank you for your information. I really appreciate you taking the time out at the end of a long day to give this valuable information. So I really thank you for your participation. And it's been a great experience. And you are all so impressive. I'm honored to work with you. So thank you very
Dr.
much.
Sharbrough. Thank you for organizing all of this and pulling it together. It was so needed by our community and us.
I agree.
Thanks, Joel.
All right.
Thank you.
Thank you.
Bye-bye.
Bye.
All right, everyone, stay safe and be well and get vaccinated.
Yes.
Getting mine on Sunday, my first.
Good.
Yes. Nice.
Good.